Yeah, hi. Today we will see the commitment, which is a part of basic concept of development of an embryo. So, commitment as an engagement or involvement that restricts freedom of action, and it can be divided into two stages. First one is the specification, and then second one is the determination. So, in this video, we will see the detail of specification. So, it can be subdivided into autonomous specification, conditional specification, and the syncytial specification. Autonomous specification, also known as the mosaic development, and in this it is a form of the embryonic specification in which a developing cell is able to differentiate without receiving external signals. So in this, the morphogenetic determinants, that is certain proteins or the mRNA, are placed in different regions of the egg cytoplasm and are distributed to the different cells as the embryo divides. These morphogenic determinants specify the cell type. So the Laurent Chabry was the first who described the autonomous specification. In other words, the egg cytoplasm is not homogeneous but rather contain different morphogenetic determinants, transcription factor or their mRNA, which influence the cell development. Next one is the regulative development, also known as the conditional specification. So, in contrast to autonomous specification, this type of specification is cell and extrinsic process that relies on cues of interaction between a cell or the from concentration gradients of the morphogenes. So, in this, if a blastomere is removed from an early embryo that uses conditional specification, the remaining embryonic cell alter their fate so that the role of the missing cells can be taken over. So, this ability of the embryonic cells to change their fates to compensate for the missing part is called as the regulation. And this embryogenesis is called as the regulative development. For an example, we can say that if a back cell was removed and transplanted into the belly region, the new forming tissue will be a belly tissue. And this result is seen because the surrounding cells and tissues influence newly forming cell. So here is the figure of the normal development of tissues and the back cell, which is removed by and transplanted into the belly region. Third one is the syncytial specification, and this type of specification is a hybrid of the autonomous and the conditional specification that occurs in insects. So, in early embryo of insect, the nuclei divide within the egg, but the cell doesn't divide, and many nuclei are formed within one common cytoplasm called as the syncytium. So in this case, the morphogenes can influence nuclei in a concentration dependent manner, and it was discovered that the cellularization of the blastodome took place either during or before the specification of the body region. So the interaction of the nuclei and the transcription factor, which eventually result in a cell specification, takes place a common cytoplasm. There may be the autonomous, conditional, and the syncytial differences among them. So in this autonomous, the specification by differential acquisition certain cytoplasmic molecules present in it, wherein the conditional specification by interaction between cells, and the syncytial specification of the body region by interaction between cytoplasmic region prior to the cellularization of the blastodome. So here are some more differences among the autonomous, conditional, and the syncytial specification. So in next video we will discuss the determination, the another part of this commitment. Thank you and keep watching.